Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. If you Google, does sugar feed cancer? You'll generally find two extremes. One will say that sugar is a poison and directly fuels tumors, and the other says diet and sugar has nothing to do with cancer at all. Both are oversimplified and wrong. Today, we're going to walk through the actual science and what it shows. So without further ado, let's get into it. So as the slide says, does sugar feed cancer? What the evidence really says. So I'm going to start by asking the National Library of Medicine, or PubMed, a question. How many publications mention the Warburg effect? And I found that 5,731 papers mention the Warburg effect. The question is, what is the Warburg effect? This paper from January of 2015, titled The Warburg Effect in Tumor Progression, Mitochondrial Oxidative Metabolism as an Anti-Metastasis Mechanism, it says here in the abstract, compared to normal cells, cancer cells strongly upregulate glucose uptake or sugar uptake and glycolysis, the breakdown of glucose, to give rise to an increased yield of intermediate glycolytic metabolites and the end product pyruvate. Moreover, glycolysis is uncoupled from the mitochondrial tricarboxylic acid TCA cycle and oxidative phosphorylation OXPHOS in cancer cells. Consequently, the majority of glycolysis-derived pyruvate is diverted to lactate fermentation and kept away from mitochondrial oxidative metabolism. This metabolic phenotype is known as the Warburg effect. It later in the paper says, by contrast, most cancer cells show conspicuous alterations in glucose metabolism. Compared to normal cells, cancer cells typically exhibit drastically increased glucose or sugar uptake and glycolytic rates, the breakdown of glucose. Increased glucose consumption generates more intermediate glycolytic metabolites and significant amount of ATP from glycolysis. Moreover, a substantial fraction of glucose carbon in the form of associated glycolytic intermediates is shunted into multiple biosynthetic pathways instead of giving rise to pyruvate. Finally, following glycolysis, most pyruvate is converted to lactate in the cytoplasm by the action of lactate dehydrogenase LDH and secreted rather than being oxidized through mitochondrial metabolism. This occurs in the presence of sufficient oxygen to support mitochondrial respiration. The metabolic phenomenon was first described by Otto Warburg and is referred to as aerobic glycolysis, or the Warburg effect. Although human cancers display a diverse range of metabolic profiles, the Warburg metabolic phenotype is a widespread cancer-associated trait. Indeed, enhanced glucose uptake by cancer cells has become a basis for positron emission tomography, PET, with 18-fluorodeoxyglucose, FDG, which preferentially accumulates in tumor cells as a result of their rapid uptake of glucose. Because of the prevalence of the phenotype, PET is an effective clinical imaging technique to detect most cancers and monitor therapeutic responses. So that was a lot, but we're going to go over kind of what that means in pictorial format based off of this graphic right here. And so we have a normal cell here, and we have not talked about metabolism in this basics course, but without saying, one of the fuels to normal cells is glucose or sugar. And so sugar has to be broken down into other chemicals before it can be used for cellular energy. The process of breaking down glucose is called glycolysis. And the end product of glycolysis is pyruvate. And when pyruvate is broken down from glucose, it will then, in normal cells, get into the mitochondria to make energy. However, what this paper is saying, and about 5,000 other papers are saying, and what PET is based off of the fact that cancer cells will uptake glucose at a much higher rate than normal cells, and they will utilize glucose at a much increased amount and rate compared to normal cells. And instead of pyruvate going into the mitochondria to make energy, what it does is pyruvate is being fermented into lactate. We'll talk about this in much greater detail later. But the title of this video is, does sugar feed cancer? And when you see graphics like this in the literature, and you see a normal cell having a very thin line here, and cancer having a very thick line here, signifying that there is a much higher utilization of glucose than a normal cell, then that pretty much answers the question. The next question that you may have is how much more glucose does a cancer cell use compared to a normal cell? And that's answered in this paper right here. In December of 2019, there was a paper published called 2-deoxy-D-glucose and its analogs from diagnostic to therapeutic agents. And I'm going to highlight this area here. Higher glucose utilization by tumor cells require overexpression of these GLUT transporters, which is not important right now, to increase glucose uptake by 20 to 30 fold. That's 20 to 30 times more compared to normal cells. And as talked about in the first paper, the entire science of FDG PET scans used to detect tumors is based on this science. It says FDG correlates with the glycolysis or the breakdown of glucose levels and is generally much higher than that of normal tissues in many common malignancies, including breast, lung, and colon cancer. And then in the paper, it'll show how this FDG glucose is being concentrated in tumor tissues, and that's how the radiologists are able to know what is normal tissue and what is cancerous tissue, because these tumors are bringing in that radioactive glucose 20 to 30 times more than the tissues around it. So the answer is yes, glucose does feed cancer. It is not an internet myth, and it is the basis of the mitochondrial metabolic theory of cancer, which we will be going over in small chunks in this basic series. If you like videos like this, please like, share, and subscribe. Until next time.